Hi, everyone. So my name is Zoe Clarke. I'm a PhD student in Gary Bader's lab at the University of Toronto. And I'm looking forward to telling you a bit about something that I've been working on, which is that MALAT1 expression consistently indicates cell quality in single-cell RNA sequencing. So as we all know, single-cell RNA sequencing is an incredible piece of technology that has been rapidly informing us about the human body. So there are some consortia out there as well, like the Tabula Sapiens Consortium, that aims to identify and characterize all the different cell types in the human body. And you can see, so there's like Tabula Sapiens came up with this map of the cells in the human body that you can see on the right there. So, you've got all these cells grouping together based on the similarity in their transcriptomes and colored by cell types. So there's many, many different cell types in the human body that they've been working on characterizing. And part of why this is all possible is because of the high throughput nature of especially droplet-based single-cell RNA sequencing. And just to give you a little reminder of exactly how droplet-based single-cell RNA sequencing works. So essentially, you have a tissue, and the cells in that tissue are dissociated. And then you have these beads that have a cell barcode associated with them. So you can see these beads as the big multicolored dots being fed through this tube here. And these beads are matched with the cell. So the cell is that little white dot, and they're matched at a bit of a low frequency rate so that you don't have multiple cells ending up in a single droplet. So you end up with all these droplets, and each droplet should have either just a bead or it has a bead and a cell. And then all of the reads in each of these droplets are paired with that cell-associated barcode that's in the droplet, as well as a unique molecular identifier. So that's how you know what's a unique read and what cell is it coming from, because then all of this cDNA is pooled together and collected in a single data set. So at this point, we've got all these cells and we've got all these droplets. They're all captured together, and we have to sort out, okay, what are cells and what are droplets? So this can be done by looking at a knee plot, by looking at the UMI counts of the cells. So what you can do is you can take all the droplets that were captured in this experiment. You can organize them from the most UMIs, so that representing the number of reads that were captured in a droplet, to the lowest number of UMIs. And what this will look like on the plot is you've got UMI counts on the y-axis, and all the cells line up on the x-axis. And so you've got these high UMI cells, lots of high UMIs, and then you have this steep drop off, and then all of a sudden you have very low UMIs, and that represents the transition from cells to droplets, empty droplets. And so you can take a look, okay, where does this steep cliff happen? Let's find the inflection point, and we can draw a line right across here and say that, okay, everything above this point is likely cells, and everything below this are likely empty droplets. However, what can happen here is then you have some low UMI cells that are just naturally this way biologically. These could be small cells or stem cells, and these may get lost and labeled as empty droplets in the experiment. So to combat this challenge, empty drops is a tool that was created that uses the background RNA expression in the experiment to rescue these low UMI cells. So essentially, empty drop says, okay, all these droplets, they're going to have little bits of this ambient RNA in them. Let's figure out this kind of average ambient profile. And then any droplet that has a transcriptome that really stands out from this profile is likely a real cell, and we should rescue that. So that really helps preserve those low UMI cells. And then following this, some additional filters may be applied. So every once in a while, multiple cells do get captured in a single droplet. So there's doublet filters that can look for those and filter them out of your sample. And then there's high mitochondrial content filters as well. So essentially saying, okay, if you have a cell in a single cell RNA-seq data set and it's got a ton of reads coming from the mitochondria, this cell is probably stressed or dying in some way. You probably want to either flag or remove those from your sample. So a lot of the time when doing this QC, you can flag these cells and visualize them on your UMAP to see how many were affected. So you can see, like in this example, everything in blue has passed the filter, everything in red has failed the filter. You tend to get rid of everything in red and keep the things in blue. And at this point, a lot of the time filtering ends here. However, I believe that at this point in a lot of different data sets, many droplets are still being captured and analyzed like real cells. So just for an example, there was this human kidney atlas that was published in Science in 2019, and the cells on the left are colored generally by what type of cell they are. So you've got cells, immune cells, some from the nephron, vasculature, and stroma. And I believe that everything in red here is actually likely an empty droplet or a poor quality cell being analyzed like a real cell. So this ends up being more than half the sample, and a lot of it's coming from the kidney nephron. And I'll tell you why I think that is. So I started to think that there's actually a lot of empty droplets in a lot of different data sets when I started looking at single cell data sets through the lens of this QC tool called Droplet QC, which improves the identification of empty droplets and damaged cells in single cell RNA sequencing data. So essentially, how Droplet QC works is it uses the splicing ratios in the cell of your single-cell RNA-seq data to determine cell quality. And Droplet QC does this by calculating this metric called the nuclear fraction for every cell in the data set. So the nuclear fraction being the fraction of unspliced nuclear RNA in a cell. So just to think back to that droplet-based single-cell RNA sequencing protocol, essentially what can happen is you can have cells that tend to be maybe a bit fragile or larger or whatnot, but their cell membranes can rupture in the experimental protocol and release a lot of their cytoplasmic RNA into the environment as ambient RNA. And then you can have droplets that take in this cytoplasmic RNA. And so what happens is you have these droplets that are filled with all of this spliced contaminating cytoplasmic RNA, and these cells would have a very low nuclear fraction. So very few unspliced reads found in these cells. And then on the other end of that, you can have these droplets that maybe have just a nucleus in them because their cell membrane has ruptured and they've lost a lot of their cytoplasmic RNA. And these have a very high nuclear fraction. And so real cells would be somewhere in the middle of these two extreme values. So just to reiterate, it's this cytoplasmic RNA that can be released and contaminating the data set. And Droplet QC can calculate the nuclear fraction and help make this clear in the data set. However, Droplet QC is computationally intensive because it requires the use of these large BAM alignment files. So on a single sample, it can take up to half an hour to an hour to run this on like a fairly small sample. So it's a bit prohibitive that way. So what I found is that MALAT1 expression is actually highly correlated with the nuclear fraction. So what you're looking at here is a tabula myricenis pancreas data set, so mouse pancreas, and you've got the UMAP up here, and on the left, UMAP MALAT1 expression is projected onto the UMAP, and on the right, you've got the nuclear fraction. And just visually, you can see that they look fairly correlated. You can see the cluster in the bottom right especially has very low MALAT1 expression values. In some cells, MALAT1 isn't expressed at all, and these cells have a very low nuclear fraction as well. So MALAT1 is a long non-coding RNA that's retained in the nucleus, and it's expressed ubiquitously across all cell types that have a nucleus. So essentially, if you don't see it, you're probably don't have a nucleus. And I found when correlating MALAT1 expression, normalized MALAT1 expression to the nuclear fraction, I looked at 16 different both single sample and integrated data sets. So I ended up looking at over 100 different samples, over 200,000 cells, and found an average correlation coefficient between the two values to be 0.71. And in 81% of the data sets, MALAT1 is just the top correlated gene with the nuclear fraction. So when looking at MALAT1 in a bunch of different data sets, what you see most often if the data set looks high quality is you'll see this normal distribution of MALAT1 in the sample. So you're looking at a couple of histograms here. The one on the left is representing a fairly high quality looking sample. You've got MALAT1 having this moderate to high expression, this very normal looking peak. And then you have a few values that are getting lower, close to zero, potentially representing a few empty droplets in the data set. And so that was the mouse adipose data set, so also from tabula muracenis. And then on the right, we're looking again at that pancreas sample, and you can see it does have that normal distribution of MALAT1, that's that kind of moderate to high peak, but then it also has this lower peak close to zero, and also a ton of zero values showing cells that aren't expressing MALAT1 at all. And I'm just highlighting those there. So essentially, we can use MALAT1 expression directly to flag or filter out these low quality cells. So I just made a function that if you provide a vector of just your normalized MALAT1 expression values, this function will plot a histogram and fit a density function to it. So the density values are what you can see just in the plot on the right. There are all those dots there. It essentially is a function that just follows the histogram. And then this function finds the local maximum that represents that expected good like MALAT1 peak, and then finds the closest local minimum to the left of that peak. And essentially, you find the distance between the local maximum and the local minimum, project that on the other side as well, isolate those density values, and just plot a quadratic to that function. And the x-intercept on the left is essentially this minimum threshold saying, okay, if you have MALAT1 above this value, this is probably a good, high quality looking cell, and anything below this MALAT1 expression value may be low quality or should be flagged to look at further as it may be an empty droplet. So essentially you can see that in this data set, right in the middle there is the histogram. I've just drawn a line right there where the MALAT1 threshold is. On the left is everything that failed the filter. On the right is everything that looks good and passed the filter. And then you can see in the UMAPs on the right as well, I use droplet QC. I use their function to flag what they think are likely empty droplets. And then I use the MALAT1 threshold as well. And you can see that it's basically the exact same cells that are being flagged and filtered from the data set. So again, just these cells that have this low MALAT1 expression, it indicates they're likely an empty droplet or a droplet that maybe has like a fragment of a nucleus in it, or is a red blood cell. And this is a very, very fast, easy, and informative thing to do. So the code for this function is available just at the GitHub link there that you can also find by scanning the QR code. The repo was just made last week, and I just submitted the manuscript last night to bioRxiv. So it's very, very recent, and more details should be up soon, but I encourage you to go take a look and check that out if you're working with single cell and you're interested. So again, just to reiterate, even just glancing at MALAT1 expression is a really fast and effective way to interpret cell quality in most single cell RNA sequencing data sets. Droplet QC, it's great, but it does require BAM files and is computationally intensive. You can't always find BAM files online if you're looking at publicly available data, and it's computationally intensive and can take hours to run. However, it does work on species that don't have MALAT1 or long non-coding RNAs annotated on their genome, so that's a good thing. Whereas the MALAT1 threshold, it does require MALAT1, but all you need is your gene expression matrix. And then also, if you're just looking at like an interactive single cell RNA sequencing data online, you can just type in MALAT1, just gives you a really general idea of what you're looking at or if there's anything that may be problematic in the data set. So after looking at this for a while, I started to think that many biological patterns that we see in a lot of different data sets may actually be technical. And so I've just got a bunch of different single cell maps here. So you've got some from kidney, tongue, heart, liver, and brain. You can see that kidney data set I showed you earlier in the bottom right. And these all just have MALAT1 expression projected onto the data set. So anything that's kind of shining bright green might be a bit of a red flag there. And I've labeled those different cell types. A lot of the time I do see these same cell types coming up as repeatedly having this low MALAT1 expression, indicating there may be some experimental quality control that could help the quality of these cells being analyzed in the data set. And this just tells me that I think we still have a lot of work to do when it comes to just generally describing and identifying general cell types. So if I bring back this tabula sapiens map from the very beginning of the presentation, and I just look at MALAT1 expression on this data set, you can see that there are certain clusters from certain tissues that are really lighting up as like potentially there's something going on here that we need to look closer at. And so this makes me wonder, what kind of technical patterns will we continue to find? So just by looking at MALAT1, it looks like large proportions of data sets may not be real whole cells in the single cell RNA sequencing data. And then looking in the literature, MALAT1 has also been found to be associated with cancer and the cell cycle, but it's also a very dominant technical signal. So when we're looking at MALAT1 expression, when is this biological and when is it representing something technical? I think it's an interesting question. And also, I think the work with Droplet QC shows how interesting splicing can be to look at in single cell RNA-seq from a technical perspective as well as a biological perspective. So, this shows that ambient RNA may be confounding RNA velocity results, for example, that rely on splicing information. And also knowing that it's cytoplasmic RNA, so spliced RNA, that can be really problematic in terms of ambient RNA in a sample. Maybe this can help us find a new ambient RNA correction method that relies on the splicing information. So, and with that, I just want to thank you all for listening, and to ISMB for having me come here and talk today. Of course, my entire lab and all of our collaborating labs and all of our sources of funding. There's the MALAT1 GitHub repo QR code if you're interested and you didn't have time to scan it before. And I'm very happy to take any questions. Thank you so much.
Great talk. Any questions? Oh, wow, lots of questions.
Thank you for very nice talk, and this is very important information for single cell analysis. And I think MALAT1 is very mysterious gene. It's often increased with status of cell, like aged cell more highly express MALAT1, or some, you showed the disease cell also highly express MALAT1. Have you ever compared this MALAT1 ratio across kind of the donors, status, age young, like that?
Yeah, so that's a really interesting question. And I have, I got a similar comment a little while back, especially about age, and hearing that MALAT1 is expressed more with age. And because I was looking a lot at the tabula miracenis dataset, because of their availability of their BAM files, they have these different age categories. And what was actually interesting is I found more what looked like droplets to be, so lower 0 MALAT1 expression values in older samples. I'm not exactly sure why that is, as animals age or as organisms age, maybe the cells become larger, potentially more fragile and prone to that. So it is the opposite pattern of what I've heard with aging, which is very interesting. So thank you.
One online question first.
Actually, we have 2 online questions. I think I will read one and then the other one. So the first question is asked by Haruka Ozaki, and the question is, did you evaluate your method on single-cell RNA-seq method other than 10x Chromium?
Yeah, I did. So unfortunately, not all data sets have BAM files that are really easy to do the droplet QC analysis. However, I have looked at SmartSeq2 data sets and see the same pattern with MALAT1 expression. And I've also looked at single-nucleus RNA-seq data sets. And in the single-nuc data sets, as expected, you're just looking at nuclei. You almost never see cells that have these really low MALAT1 expression values, which I think can kind of serve as a positive control. But every once in a while, you do see a single nuc cell that has no MALAT1 in it, which is very interesting. So yeah, I see this pattern across different single cell data sets.
Okay, thank you for the answer. And the second question is asked by Philip McCown. Nice talk. I was wondering, as some MALAT1 gene annotations only use a tiny section of the 3' end of MALAT1 that is highly duplicated in other long non-coding RNAs of the human and mouse genome. Did you verify that you were exclusively examining MALAT1, which might also be hampering some findings that you have in other data sets?
That's a great question. And the simple answer is no, I am relying on the callings from the single cell RNA sequencing method for the most part.
Okay, let's do 2 quick questions. Who has the...
Okay, a quick question. So any correlated genes to MALAT1 that can be used instead of MALAT1 in case that's missing for some reason?
Yeah, so NEAT1 and EXIST XIST, are a couple of ones that I see fairly correlated with the nuclear fraction as well. They're not quite as consistently and strongly expressed, and they're often not that number one correlated gene like I see MALAT1 as most of the time, but those are also genes that have found to be expressed in the nucleus. So yeah, you can look for those as well.
Okay, last one.
Thank you. It was a great talk. You mentioned that you see the empty cells with less MALAT1 in repeated cell types across the data sets. Aren't you worried that's a biological problem and not a technical problem in creating the data sets, and therefore we're missing stuff?
Yeah, definitely. And I've thought about that, and I think that's a great thing to think about. So this is where I would encourage people who are more tissue experts to really think about why is MALAT1 not being expressed there. I've mostly worked with the liver, which has a lot of quality control problems, and we do see repeatedly MALAT1 being variably or very lowly expressed in hepatocytes, which again, we know to have a lot of issues in the liver. And so when looking at these other tissues, I'm a lot less familiar with them personally. So if anything, I would love to treat this as like a, yes, please go explore, check out MALAT1 in your data. I think it's probably a technical signal, but there's always the chance it's a biological signal. And if anything, it's just something to pay attention to. So great question. And I just wanted to say as well, I'll be here during lunch. Please come talk to me. I love talking about MALAT1. It's like my favorite topic these days. So yeah, thank you so much.
Okay, awesome. Thanks.